Good afternoon, good morning, good evening to you all from wherever you are, and welcome once again to the third day of VG24. My name is Muigai, and I'll be the chair of this session together with Samuel, who will be here with us. We have had slight change in the order of the sessions this afternoon. We'll start with Katie Watson from Stellenbosch University presenting seagrass transcriptomic responses to heat stress. This will be followed by Laura Tensen from University of Johannesburg, who will give us a talk on selection drives, phenotypic divergence of Cape leopards. We'll move on to Nidal Fetnassi, who will give us a presentation on biodiversity, updates to the checklist of Lepidoptera in the Central Highlands Mountains of Morocco. And then this will be followed by a talk by Abdessamad Aglagane on applying genomics to support conservation assessments of Moroccan biodiversity, the Saharan honeybee. After this, we will have an interlude where we'll be able to interrogate our speakers, ask your questions, have some discussion. So please do put your questions in the Slido. And now I'd like to hand over to Katie Watson of Stellenbosch University with her presentation on seagrass transcriptomic responses to heat stress.
Hi everyone, today I'm excited to share with you my work exploring transcriptomic and photophysiological responses to thermal stress into genomically divergent seagrass populations. So for those of you who don't know what seagrasses are, they are marine plants capable of engineering coastal environments to create a unique habitat that provides a vast array of ecosystem services, playing pivotal roles in supporting human well-being and biodiversity. These include things like water oxidation and purification to protecting coastlines from erosion and flooding, as well as acting as a carbon sink. And this is something that seagrasses have been gaining a lot of media attention for recently, as they act as a carbon sink responsible for between 10 and 18% of carbon burial in the oceans yearly. And this is despite occupying less than 0.1% of the ocean's surface. And they can sequester this carbon for millennia at rates 35 times faster than tropical rainforests. However, despite being one of the most important and valuable habitats globally, seagrass meadows are also among the most threatened and facing the 5 most serious threats to marine biodiversity. These include direct losses through activities such as port development, whilst indirect but potentially more damaging impacts relate to water quality degradation and, of course, global climate change. With recent estimates finding a substantial net loss in seagrass area cover globally of approximately 19%, and this is only predicted to continue. So species are often strongly influenced by environmental extremes, with thermal maxima being important predictors of species extinctions. And particularly in Western South Africa, future climate change projections have highlighted this region to be a marine climate change hotspot. However, seagrass populations locally adapted to thermal fluctuations potentially exhibit higher thermotolerance compared to populations found in more heterogeneous environments. So, in this era of the Anthropocene, it is critical to gain a deeper understanding of the mechanisms driving local adaptation to thermal stress, which may provide insights into signals of resilience within seagrass populations. And this is where we can apply transcriptomics. So as a side quest, before we explored the resilience of Zostera capensis, we conducted a global review of the literature on transcriptomics applied to seagrasses. And we found that gene expression studies are really geographically concentrated, being limited to just a few species, thus presenting clear mismatches between global seagrass distribution and research output. And I want to give a quick shout out to a postdoc from our lab with the first seagrass transcriptomics paper in Africa, which I'll be talking about later. What's more, there's also the potential to begin to link transcriptomic responses to photophysiological measurements. So, for example, in Posidonia australis, previous work has shown distinct gene expression profiles, as you can see here, between the control groups in green in comparison to plants exposed to a heat wave in blue or even a secondary heat wave in red. And these expression profiles also differ between species, as shown here for Zostera muelleri. So here we can see that in the heat-stressed plants, photosynthetic efficiency is significantly reduced. This was found across both species. And if we can begin to link these photophysiological responses to the transcriptomic profiles, this could provide a less expensive in-field monitoring tool in response to thermal stress events. So this brings us to the objectives of this work, looking to explore the molecular and photophysiological responses of two genomically divergent seagrass populations to a simulated marine heat wave, and consider if these populations employ different stress responses, potentially due to adaptive divergence. So the dominant species here in South Africa is Cape dwarf eelgrass, with vast ranges extending up to southern Kenya. Yet isolated populations coupled with extreme fluctuations in meadow cover are fueling growing concerns around the longevity of this species. And as part of my work with the IUCN Seagrass Specialist Group, Zostera capensis has now been reclassified to endangered. Specifically, I wanted to look at populations within Langabon Lagoon on the west coast, where a pooled genomic approach found two genomic lineages within the lagoon, broadly corresponding to populations on the west and east coasts, with the two lineages shaped by frequency differences of outlier loci. And what's more, shared variation at outlier loci potentially indicated putative local adaptation to temperature and precipitation gradients between these two lineages. And RADSeq work has also supported this, showing strong genomic structure of populations across the coast. Within Langabon Lagoon, we find that these populations are experiencing different local conditions, with Klein Oosterwal experiencing more stable and cooler conditions, whereas Heelbeck is more thermally dynamic as a local environment. So differential gene expression analysis has previously been done on Zostera capensis to compare the responses of heat-stressed and pre-warming plants, and this was done by Andrew, our postdoc. A couple of interesting findings from this work are the upregulation of heat shock proteins, which signal heat stress. Also, most of the differential gene expression was driven by downregulation, which suggests that for this species, gene deactivation is more important than gene activation during stress response. So, for example, genes related to photosynthesis were downregulated, likely to protect heat-sensitive photosynthetic machinery. But this is really in the beginning of what is possible for us to discover with transcriptomics. So for my work, shoots were collected from the two genomic lineages within Langabon Lagoon and exposed to a simulated marine heat wave using a common garden design, where water temperature was rapidly increased to 34 degrees for 3 days, followed by a recovery period at 22 degrees. And photosynthetic performance measurements were also taken using a diving pan at each of the extraction time points across both populations and treatments. Briefly, this is the workflow I followed, which I'm not going to focus on too much now, but please feel free to contact me afterwards. I'm more than happy to get into the nitty-gritty of this with you. So onto the exciting part, the results. Although photophysiological performance declined following the marine heat wave, statistical analysis did not show significant differences between treatments or between populations. And this was found across different measures of photosynthetic performance as well, which potentially highlights the plasticity of the species to stress or the lag in photophysical response, yet the transcriptomic response was far more dynamic and responsive. So the response to heat stress was similar in both populations, with expression primarily driven by downregulation of functions such as the regulation of photosynthetic processes as expected, as well as DNA binding and translation. Yet upregulated genes included defense responses to cellular heat stress, including the regulation of heat stress proteins relating to DNA repair and replication, as well as genes involved in structural developments that can reduce yield. However, Heelbeck upregulated a greater proportion of genes relating to heat stress defense and many unique upregulated genes encoding proteins involved in acquired thermotolerance. And this may reflect a more complete heat stress response in comparison to Klein Oosterwal. Populations showed differences in the types of heat stress proteins differentially expressed, with Heelbeck exhibiting signals of adaptive defense against abiotic stress, including the upregulation of heat shock proteins, whereas Klein Oosterwald inhibited heat stress-induced senescence, which is associated with heat tolerance in plants. And then following the recovery phase, expression profiles were now driven by gene upregulation, with populations showing divergent differential expression, where Klein Oosterwald downregulated processes related to RNA binding, defense responses, and cell membrane organization, with upregulation of genes relating to reproduction that regulate fruit ripening and pollen development. As we know, seagrass reproduction is usually in response to stress. Whereas comparatively, Heelbeck downregulated functions relating to photosynthesis, likely as a mechanism to prioritize upregulation of genes that enable the repair of unfolded denatured proteins as part of heat stress tolerance. So what are the conclusions that we can take from this work? So our study demonstrates the effects of thermal stress between these two genomically divergent seagrass populations, and this provides us with insights into the intraspecific thermal resilience of this species, with results suggesting that for Zostera capensis, there is a great deal of plasticity to acute heat stress, and we also found evidence to support local adaptation. But both populations did show mechanisms associated with recovery, with potential implications for climate resilience focused restoration efforts. And this is where future research can really focus, looking at stress memory or thermal priming. So keep your eyes peeled for this publication, which is currently in review and should hopefully be out soon. And with that, I would like to thank my wonderful collaborators and funders who've made this work possible. And I would now like to welcome any questions. So please feel free to contact me via email or Instagram or Twitter. Thank you so much.
Hi everyone, I'm Laura and I'll be talking about divergent selection in leopards from the Cape of South Africa. So a short introduction to South Africa. It's a country of many extremes. It's the 6th most mega diverse country in the world. There are 7 to 9 biomes depending on how you define biome. It's home to 10% of the world's plant species and 7% of the reptile, bird and mammal species. And 6,000 species are endemic to the fynbos biome alone, which occurs in the very southern tip of Africa. And there are also 4 kinds of leopards, and I'm going to explain now what I mean by that. So there was a while ago, there was a study by Anko et al. He looked at NDH5 genes from ancient DNA museum samples, and he found 3 clades across Africa. So that's West Central Africa, it's Central Africa, and Southern Africa. And you can see that the Central African and Southern African clades seem to meet in the northern part of South Africa. So we thought that was an interesting study area. So we collected some samples and did a first mitogenomic characterization of this population. And what you can see in a phylogenetic tree, that actually these clades are very deeply diverged. You can see at the bottom, all African samples. So this is one subspecies, but you see these deeply diverged clades that is deeper than the divergence between Asian and extinct European leopards. And you can also see that the Asian subspecies are actually very hard to tell apart looking at mitochondrial DNA. But most importantly, these deeply diverged clades that occur in one area, they're part of the same population. So we measured that the split occurred about 800,000 years ago, which was during the climatically unstable Pleistocene. And we predict that this area has became isolated during this time period. If we now look at whole genomes instead, we see a completely different pattern. So here we put our South African leopards into a population data set made available by Peknerova et al. And then if you look at SNPs, so these are neutral, normally SNPs and indels, you can see a very clear isolation by distance pattern, where only Ghana seems to be an outlier. Whereas if you look for structural variants here at the bottom, you don't see any of this structure at all. So in an admixture plot, you can see again that these leopards in northern South Africa seem to be forming a sort of admixture zone between leopards from Zambia and Namibia. And yet again, it's important to emphasize that this is all one subspecies, although there is some population structure. So the leopard number 3 would be the red or strawberry leopard. This occurs in the same area. It's a color variant. And we were curious why this leopard seems to be increasing in numbers currently. So we again sequenced the whole genome. First of all, we found that a deletion in the TRP1 gene seemed to be causing this rare color variant. And there seems to be higher levels of homozygosity in this population. So we think that genetic drift is locally overruling natural selection as a result of this high illegal uptake that seems to be happening in the area. So again, on your left, you can now see the Central African clade to which the red leopard belongs. In this case, we have the Southern African clade, but there's another leopard. And that one occurs in the Cape of South Africa, here at the very bottom in that map. And this leopard is quite special because it's morphologically distinct as well. It seems to be almost half the size of leopards elsewhere in Africa. So it's become quite a legend as well. There's the Cape Leopard Trust, the Cape Leopard Wine, and there's a lot of conservation efforts on the ground because it's a very fragile population. So people have suspected why it might be smaller in size. So the prey size seems to be a lot smaller in this area compared to the savanna biome. So at the bottom, you can see leopards from Mpumalanga with an average body size of about 50 kilograms. In the Cape, it's about 28. And the prey size is also much smaller. And as a result, the home ranges of leopards in the Western Cape are much larger, around 1,000 square kilometers. So a bit of the history on the leopards. So it originated about 2 million years ago in Africa. We found that deep divergence, mitochondrial divergence, about 800,000 years ago. 600,000 years ago, there was an out of Africa dispersal event which caused all these subspecies that now occur in Asia. And then interestingly, about 24,000 years ago, there was a diversification event in the Cape of South Africa. Because before this, the area was very dry, almost desert-like. But as a result of these cold to warm oscillations, the area became much wetter, and that allowed a lot of species to invade, plant species and small mammals. But a lot of large herbivores didn't make it to the area because the soil is still very dry. Also, what's important is that in the 1600s, colonization events happened, and this led to the extinction of many local species, such as the bluebuck and the guaca and the Cape lion, and the Cape leopards took a hit as well. So a little bit more about the Cape leopard then. It has been suspected to be a subspecies because it's morphologically different. It went through a population bottleneck of about 50 individuals during this colonialism. Currently, there's about 200 to 600 individuals. There's strong habitat fragmentation and urbanization in the area. And as a result, Cape leopards have now mainly retreated to this mountainous, rugged terrain. But gene flow does appear to occur based on microsatellite analysis and based on camera trapping data. So then we wondered whether leopards from the Western Cape are genetically distinct, what their demographic history is, and whether there are signatures of selection that underpin genomic and phenotypic divergence. So to quickly go back to our map, we previously had that study in Northern South Africa. We now added 10 whole genomes from the Western Cape, and we also got some genomes from Namibia, from NCBI. So we compared these 3 populations. Some of the methods we used were NGS at mix, PCMC, and GON to look at these effective population size, both historic and recent. From the PCMC runs, we deducted this MISTI, which you can use to measure divergence between populations, and we did cell scan to look for signatures of selection. So here's a bit on genetic diversity. We found that nucleotide diversity wasn't high, nor was it low, and primarily that it doesn't seem to be any differences between the populations. The same goes for the runs of homozygosity, which does not seem to be alarmingly high in the Western Cape compared to other areas, which would sort of support the idea that this population isn't isolated. So we do find population structure, though, very interestingly. So the Western Cape population clearly distinguishes itself on the PCA, and we see on the right that the highest likelihood for the number of clusters was 3, where where the Cape leopards are clearly distinct from Mpumalanga and Namibia. So the genetic distance between Namibia and Mpumalanga is about 0.03, which is to be expected based on the distance between them. And with the Cape leopards, that's much higher. It's about 0.07. So then we put the Western Cape and Namibian leopards into that phylogenetic comparisons that we did previously. And you can see that the Namibian leopards clearly cluster with the Western Cape, although there's still about 100 base pair differences between them. So on the left, you can see the haplotype network. This is only protein coding genes. So albeit they cluster together, the divergence between them is still quite substantial. And you can see that the Mpumalanga cluster has about 200 base pair differences. So then based on the PSMC, we found that the effective population size greatly reduced about 500,000 years ago. So this was both in Mpumalanga and the Western Cape. But then if you use MISTI to measure divergence time between them, we did see that the population started diverging about 24,000 years ago. And if you do the same calculations for a Cape Namibia comparison, this was about 12,000 years ago, which probably might relate to the fact that these populations are part of the same mitochondrial cluster. So on the right side, you can see the effective population size more recently. And you can see that there was a great reduction from about 6,000 leopards to barely any shortly after British occupation. And this is very likely to be related to an institutionalized bounty on leopard's head that was put in place that lasted for almost 100 years. Then we looked for signals of selection. So for enriched genes, we found 102 genes that were under positive selection in the Cape leopards compared to the other populations. Most of these seem to relate to like skeletal development or metabolism, but some genes were picked up that are actually quite interesting. For instance, we have the AC1, which is related to unsaturated fatty acid oxidation. We have the PLIN4, which reduces cardiac lipid metabolism. We have GPSM1, which is related to accumulation of fat tissue. We have ACOX3, which increases fat storage and represses fat burning during fasting. We have LSC39, which is a critical non-compensatory role when zinc is deficient. And we have STRAS6, which transports vitamin A when faced with zinc deficiency. So if we go back to our map, like I told you, the Western Cape is characterized by the pine boss biome that I mentioned earlier. And what's also important that although this is like a biodiversity hotspot, this is mainly plant species. And there does seem to be a lower abundance of large prey, and certain species have never managed to settle in the area because of the arid conditions. The soil is still very dry. And currently, like I said, leopards have retreated to these mountainous rugged terrain, which are normally characterized by lower prey densities as well. So if you compare the two areas, besides that the prey diversity and density are much higher in the north, in the savanna biome, the climate is also much harsher in the south. So very strong seasons. So it's very well possible that all these genes that we found under positive selection in the Cape relate to this nutrient scarcity, these seasonal changes in nutrient availability or prey availability. And it might explain why they're smaller in size as well. So to conclude, we found these unique morphological traits and an ecotype association. We found mitochondrial and nuclear DNA genome-wide population structure. We found that the Cape leopard diverged 24,000 years ago, or 12,000 to 24,000 years ago. And we found signatures of selection that suggest local adaptation to nutrient scarcity in the area. If we then go have a look at how IUCN, the CAST specialist group, defines a subspecies, which is a group within a species that mostly share morphological and molecular characteristics that occupy a distinct part of the geographical range, although gene flow is also expected between subspecies where ranges are continuous. So that's definitely something to keep in mind. And I think it calls for more in-depth analysis of this unique leopard population in the southern tip of Africa. Thank you very much, and let me know if you have any questions.
Ladies and gentlemen, esteemed colleagues and fellow researchers, my name is Nidal Fetnassi, a PhD student at Tartu University of Estonia and Cadi Ayyad University of Morocco. It is my great pleasure to present our recent findings on the Lepidoptera of Morocco, specifically focusing on the Macrolepidoptera of the Central High Atlas region. Speaking of the region, the High Atlas Mountains stretching across North Africa stand as a region of remarkable biogeographic diversity and form a crucial part of the Mediterranean's biodiversity hotspot. The High Atlas Mountains are crucial for ecological balance, providing habitats for diverse flora and fauna. Due to their exceptional position between the Mediterranean Sea and the Sahara Desert, These mountains create a variety of environmental conditions supporting a wide range of species, many of which are endemic. Despite the ecological significance of the High Atlas Mountains, certain animal groups, especially insects, have received little attention. While Moroccan butterflies have been extensively studied, nocturnal Lepidoptera remain underexplored. The most comprehensive catalogue of Moroccan moths was published by Charles Rungs in 1979 and 1981, but since then there were no inventories published. Recently, most studies on Moroccan moths has primarily focused on descriptions of new species or genera and taxonomic revisions of existing groups, or in the context of applied sciences such as pest management. However, despite all these efforts, there is no comprehensive updated catalogue of Moroccan moths combining all recent discoveries. Thus, our aim was to provide an update to the knowledge of Macroheterocera fauna of the Central High Atlas Mountains with the ultimate goal of enhancing, first of all, the understanding of macromoth fauna of the region, but also establishing a basis for advancing research, conservation, and management initiatives. So our study was conducted in the High Atlas Mountains, Central High Atlas Mountains, in 9 localities of the same region from 2021 and 2022. And each of the locality was characterized by 3 habitat types. First habitat type is natural habitat, represented by forests. Second one is habitat, represented by riverbanks. And the third one is the agricultural lands, represented by mainly olive, apple plantations, as well as herbaceous field crops. As a method, we use the sugar bait trapping technique, which consists of a luring agent placed on the top of the trap, made of a mixture of red wine with sugar, and the killing agent made of ethyl acetate mixed in a bucket in the bottom of the trap. As for more preparation, once we empty the trap and in the lab, moths were stored in specific envelopes to prevent the moths from being damaged and preserve the scales. As for identifications of the specimens, moths were first of all identified by external morphology. In case when morphology was not very successful, we conducted genitalia dissections. And again, when both techniques did not yield any conclusive results, as some of our traps were completely damaged by mantids, part of the wings of the abdomen structures were missing, or in the case when the species were poorly known, we then proceeded with DNA barcoding. A short overview of about how DNA barcoding was conducted in our case. First, legs were carefully collected for DNA isolation, followed then by PCR amplification and then sequencing. Once we got the sequences, we submitted them on NCBI database to determine their corresponding species. How many specimens were barcoded versus how many specimens were mounted or dissected? So almost 90–70% of our specimens were identified based on traditional techniques. However, 2.4% of our specimens were barcoded, representing 110 individuals. As results, so we collected a total of 4,554 specimens of Macroheterocera, representing 125 species, belonging mainly to the following families: Noctuidae, Erebidae, Geometridae, Drepanidae, and Euthalidae. Among our findings, we found 3 endemic species to Morocco, as indicated by Rungs 1981 as the only national reference we have. These species are Alophias powelli, Apamea marocana, and Kersutis rungsi. Some very important findings: we found 2 new species for Morocco. These species are Exoacos and Polymyxis argilosiago. And again, comparing our results to Rungs catalog 1971 and 1981, we found 13 species that were previously reported from other regions of Morocco, but they had not been reported from our study area. These species are Polypogon plimigralis, Scoliopteryx lebatrix, Catocala elocata, Catocala oberturi, Clytia illunaris, Acronicta psi, Graniophora pontica, Tita luctuosa, Lepidoptera cilium, Paradrina flavirena, Lucani ezeai, Ixoa temera, and finally Ocroplora leucogaster. Some other findings. We also found 7 species that were not earlier mentioned by Rungs 1981, but they were later reported by other foreign specialists that have shortly visited Morocco. These species are Opitacia cuptata, Clytia infrequens, Cryphia lusitanica, Paradrina cadeni, Biotyp occidentalis, Mitimna languida, and Mitimna congrua. We could actually not successfully determine the following species after conducting DNA barcoding. These species actually remained at the genus level even after DNA barcoding. So 4 specimens remained unidentified and they belonged to Cryphia subgenus Otalis espenia pallida, 1 specimen of Cryphia subgenus Cryphia espenia simulatricula, 2 specimens of Antarchaea espenia erubescens, 1 specimen of Polymexis espenia subvenusta, and finally 1 specimen from Caradrina espenia salini. We think that this could be due to several reasons. First of all, maybe there is a lack of reference sequences on the NCBI database, but we also thought that these maybe could be issued from hybridization, but even more, we thought that these could be new species to Morocco. So we are at the moment thinking about performing more investigations to find out what they could be. Okay, again, to sum up, we found 125 species in the central high Atlas region of Morocco belonging to the following families. And among these species, 2 species were new to Morocco, 13 species were new to the region, 7 species were reported later, but they have never been described earlier by Rungs 1981. And 5 species remained at the genus level. And this possible reasons behind it is that there is a lack of reference sequences. Probably these species could be issued from hybridization, or maybe they could be new species to Morocco. So we concluded that, of course, DNA barcoding seems to be a powerful tool, but when dealing with damaged or unidentified specimens, it is crucial to integrate both traditional and modern techniques. As these combined approaches, they increase the accuracy and reliability of specimen identification, especially when they are damaged or they are poorly known. And yeah, I would like to address my thanks to my collaborators from both universities, Professor Thomas Tamaru and Dr. Erki Onap from Tartu University, and Professor Muhammad Ghamizi and Dr. Omar Gidi and Dr. Abdessamad Aglagane from Cadi Ayyad University. And thank you for your attention.
Hello everyone, my name is Abdessamad. I'm a researcher at the University of Eziuzor, Morocco, and today I will be talking about applying genetics to support conservation assessments of Moroccan biodiversity, the Saharan honeybee, as a case study. But before we delve in, I would like to give a general overview of the Apis mellifera species distribution and population structuring. So the Western honeybee exhibits some multifaceted nature as a native or exotic species. The natural distribution of the species is depicted here in blue, and it covers Africa, Europe, and Western Asia. However, with the high ecological and economic importance of this species, it is now artificially spread all over the world, as you can see in red. Based on morphometrics and molecular tools, Apis mellifera has been divided into 7 different evolutionary lineages, 3 in Africa, 2 in Europe, and 2 in Western Asia. And given the diversity of habitats it occupies, the Western honeybee presents a high subspecific diversity with about 31 subspecies recognized today, 11 of which are found only in Africa. Morocco presents the characteristic of harboring 3 honeybee subspecies, The first one is called Apis mellifera major and is found north of the Rift Mountains. The second one is Apis mellifera intermissa, also called the Tilian honeybee, is located between the Atlantic coast to the west and the high Atlas Mountains to the south. And finally, the Saharan honeybee, Apis mellifera sahariensis, which is found naturally in the oasis of southeastern Morocco. So the questions that comes to mind now is why the interest in the Saharan honeybee? And in order to answer that, we have to go back a little bit in time. Back in 1980s, this subspecies faced several pressures like drought, locust treatment, and the introduction of the varroa mite, which have led to a series of high mortality rates of beehives. Faced with these situations, beekeepers of the region started to import honeybee colonies in order to restore their stock. Unfortunately, these colonies represent mainly the Tilian honeybee, Apis mellifera intermissa. In addition, this period of time has known also a massive shift toward modernization, and beekeepers started practicing migratory beekeeping to look for other sources of honey. These two practices have gradually led to hybridization and change in the genetic pool of local subspecies, as well as the introduction of pests and pathogens which ultimately pose a serious threat to the indigenous subspecies Apis mellifera sahariensis. Thus, the study was carried out to investigate the conservation status of the Saharan honeybee in its natural range of distribution, with the aim of evaluating its genetic diversity and quantifying the levels of introgressive hybridization with other exotic subspecies. In order to address those aims, we have sampled honeybees representing both the Saharan and Tilian subspecies, and we used geographic coordinates, annual precipitation, and annual mean temperature to cluster them into seven different populations. We also used two European subspecies, Apis mellifera and Apis mellifera carnica, as reference material. The first part of this study was dedicated to population genetics, in which we calculated a bunch of genetic parameters such as the main number of alleles and allelic richness. We also measured the genetic differentiation between the studied population using the pairwise F statistics. This is a summary of the results that we have obtained. The graphs here show allele accumulation curves and estimated average allelic richness, respectively, for the populations studied. Both of these plots show that Moroccan honeybees exhibited higher and statistically significant genetic diversity over European subspecies. The same trend was also shown when comparing Moroccan honeybees with each other, where populations of the Saharan subspecies show higher genetic diversity over the Tilian populations. However, this difference was not statistically significant. Results of the genetic differentiation between studied populations resulted from the pairwise FST matrix have revealed weak to moderate divergence between Moroccan populations. However, when Moroccan populations were grouped by subspecies and hybrids were excluded from the analysis, we found a moderate differentiation between Apis mellifera sahariensis and intermissa. On the other hand, both Moroccan subspecies showed high significant differentiation from both European subspecies. We were also interested in studying the population structure and the mixture of samples, and we used two clustering methods. The first one is structure, and the second is discriminant analysis of principal component. The two methods were used as complementary approaches to infer population ancestry, evaluate gene flow between populations, identify the most probable number of clusters, and finally, evaluate the levels of introgressive hybridization between populations. The first clustering method has identified that the most probable number of clusters for the studied populations is 4, as supported by both parsimony index and the delta K values. When looking at the number of clusters equals to 3, we can identify the traditional structuring of our samples into evolutionary lineages, where the Moroccan samples were grouped as part of the African lineage. When setting the number of clusters to 4, we see that the Moroccan honeybee populations were assigned to 2 clusters. One cluster in red predominantly comprised samples from north of the High Atlas Mountains, which could be interpreted as Apis mellifera and Termissa, while the other cluster in green was more common in samples from Saharan populations and identified as Sahariensis. The graph also shows signs of hybridization between the two Moroccan subspecies, especially within the Saharan honeybee, while no significant integration of European subspecies was detected. The DAPC also revealed 3 to 4 clusters as the best model describing the population structure. The 3 groups indicated a deep subdivision into evolutionary lineages A, M, and C, while the 4 groups indicated a subdivision into subspecies with Moroccan samples showing overlapping groups. When involving all Moroccan populations as depicted in the graph to the right, we can see that samples from West and South Saharan populations showed the least integration levels. Based on the 2 methods we used in the study, we were able to assign each sample to the subspecies of origin. First, with the DAPC method, we weren't able to identify hybrids, and samples were either assigned to Sahariensis or Intermissa. With this method, we can clearly see that the genetic pool of the Saharan honeybee has drastically changed, especially in North and East Saharan populations. With the structure method, and using a Q value cutoff of 90%, the situation seems more alarming. The majority of populations show few samples that were identified as pure Saharan honeybees, while most of samples were identified as either introgressed or pure Chilean. To sum up, this is the first study ever to be conducted in Morocco using nuclear markers to evaluate the genetic diversity and population structure of the Moroccan honeybee. We have shown that Moroccan populations exhibited higher genetic diversity over European ones, which is in accordance with previous reports showing that African honeybees are generally more genetically diverse. We also showed a moderate genetic differentiation between Saharan and Chilean populations, indicating an extended degree of hybridization that leads to a more apparent homogenization between the 2 subspecies. Finally, no significant admixture of European honeybees subspecies was found in Moroccan populations, while at the same time there is an extremely ongoing integressive hybridization between the Saharan and the Chilean subspecies, which strongly highlight the artificial sympatry of Moroccan subspecies as a major threat. With that, I have come to the end of my presentation, and I'll be happy to have any questions. Thank you very much.
Thank you everyone for joining us in this session, and thank you the speakers for your wonderful presentations. Meanwhile, we are sorry for the earlier technical hitch during the talk of Cadi. Please, if you still have questions for the speakers, kindly have them written down on the Slido. We have questions for the speakers. The first here goes to Cathy Watson. The question is, in the seagrass meadows, are the seagrasses the only organisms fixing carbon? What roles do other inhabitants of the biome play?
Sorry, could you repeat the question? I just missed the middle part.
Oh, sorry. In the seagrass meadows, are the seagrasses the only organisms fixing carbon? Maybe I take it one after the
fixing
other.
carbon. Oh, okay. And what role
Yes.
do the other? Yeah. So at the moment, we believe that the seagrass meadows are the main drivers of carbon sequestration. That's not the primary focus of my work, so I'm not entirely sure the extent to which other macrofauna are potentially contributing to climate stability through storing carbon. But yeah, the seagrasses themselves are the main drivers. And potentially there is also a relationship with surrounding coastal systems, particularly when they're found in mosaic with mangroves or coral reefs.
Okay, so the second part, even though you a little bit talked on it, is that what are the roles of other inhabitants of the biome in the biome? What roles do other inhabitants of the biome play?
So this is an interesting point. Other work that's going on in our research lab at the moment is trying to understand how much of the carbon that is sequestered by seagrass meadows is actually from the seagrass themselves, or is in relation to other surrounding marine vegetated ecosystems. So this includes things like salt marsh, mangroves, or, as I say, coral reefs. Within South Africa, the seagrasses are also found in mosaic with salt marsh habitats. And we find that predominantly from looking at stable isotope analysis, we are finding that predominantly the carbon sequestered within seagrass meadows is from the seagrass because of the carbon signature. We can determine that rather than from outside sources. But taking that in an ecosystem scale, generally mangroves, when they're found in mosaic with mangroves, are sequestering a greater proportion of carbon than seagrasses, then followed by salt marsh and then seagrasses.
Okay, thank you, Katie. The next question goes to Laura. The zinc-related genes under selection, is there a geogenic driver of high or low zinc because of the underlying geology?
Yeah, we did look into that, and we found a study that compared the zinc in herbivores, plants, and carnivores in the Cape compared to Kruger, and we did see that most animals in the Cape are zinc deficient, most strongly of which predators, and I think it's because the soil in the Cape is much drier, so it's a very sandy soil that apparently doesn't hold zinc and other nutrients very well. So as a result, you see changes in plant community as well, such as C4 grasses instead of C3. So we do think there might be a correlation to zinc deficiency, but this is something that needs a lot more investigation.
Okay. Thank you, Laura, on the Cape Town leopards. Next is to Nidal. How connected are the Moroccan and Iberian lepidopteran faunas? Do you think that some apparently widespread taxa could be instead multiple?
So yeah, I think actually that the Moroccan and Iberian Lepidopteran fauna are quite connected, given their proximity and historical land bridges, which enable species migration. But the thing is that Morocco has actually diverse climates and habitat ranging from the Mediterranean to the Saharan environments. It fosters actually some unique ecological niches, likely driven genetic divergence even in species shared with Iberia. So I guess that this variation actually can often lead to cryptic species where morphologically similar taxa actually can be genetically distant. Regarding the second question, I think that recent molecular studies increasingly reveal that some of the taxa thought to be widespread across Morocco and Iberia are in fact complex as distinct species, given that Lepidoptera has stronger habitat or specializations actually and host-plant relationships. But I think it's highly plausible that further researcher will uncover additional aquatic species between these regions.
Okay, thank you, Nidal. One more question, and this goes to Abdessamad. Of isolation by distance in the Moroccan honeybees. or has the migratory beekeeping obscured any signal of expected impact of the high Atlas barrier?
Yeah, but so we haven't tested the isolation by distance factor as a contributor to the isolation of the two subspecies. But yeah, I think with the migratory beekeeping and the beekeeping practices in the area could contribute to obscuring any signal of the expected significance of the natural barrier, such as the high atlas. But in the forthcoming analysis, we are doing whole genome sequencing analysis, and we are thinking of seeing how the isolation by distance factors into the equation.
Okay, thank you for your response. If you still have questions for them, you may put them down on the slide. We move to the second part of this session. Thank you for your responses. Next on the line, we present the talk from Nazifo Mugagula from Tshwane University of Technology, Pretoria, on recombination hotspot landscape in South African beef cattle. And this will be closely followed by Shian Mura talk from Diplomics, Cape Town, on 1KSA, decoding South Africa's biodiversity. And the last talk of this session will be from Mashish Omar from the Faculty of Sciences, Muhammad V. University in Rabat, Morocco, on structural genomics of bats endocannabinoid system. And after the presentation of their talks, it will be followed by Q&A, as we have just done from the first 4 speakers. Over to their talk, please.
Good day, everybody. I am Ms. Nozipho Makakula from the Tshwane University of Technology. And my talk today will be based on my master's project, which is titled Recombination Hotspot Landscape in South African Beef Cattle. I am not alone in this project. I'm with my supervisors, Professor Mtileni, Dr. Ngube, as well as Dr. Zwane. Now, for just a bit of background in the study, it is important to note that South African beef industry comprises of 2 sectors, that being the commercial as well as the communal sectors. Now, in these 2 sectors, we can find these 2 breeds, being the Bonsmara and as well as the Mbuni. Now, these breeds are mostly indigenous to South Africa. It is very, very important to understand that cattle play a very significant role in the livelihoods of many individuals all across the globe, but more particularly in developing countries like South Africa. Now, why is this important? Cattle play a very huge role by being the main source of income in rural areas, as well as being a means of social status in rural areas. However, both in rural as well as in urban areas, they play a very key role by being the primary nutritional requirement in most individuals, that nutrition being animal protein. Therefore, it is very important to then understand meiotic recombination that occurs in these animal species in such developing countries like South Africa. Why is this important? It is important for us to do this in order for us to better interpret patterns of variation and design better optimal breeding programs. The identification of recombination patterns and genes that are involved in recombination occurring in these indigenous breeds may help us in understanding the architecture of valuable traits that are valuable in the beef industry, in the local beef industry. Now, upon visiting literature, we then learned that recombination has been investigated in most livestock species all across the globe, and findings do show that it often does yield to traits that are of great economic importance. Those traits do include that of adaptability, production as well as reproduction. Investigation of recombination patterns has not yet been thoroughly explored in indigenous breeds of developing countries like South Africa. And by understanding the nature of recombination, the nature of valuable traits that are involved in the local beef industries that are brought by recombination, we can then better design selection programs that will favor desirable traits in the local beef industry. This gap in knowledge then presented us with a wonderful opportunity to identify recombination hotspot patterns in South African Bonsmara and Nguni cattle breeds, which this research was aimed to do. Now, the specific objective that was set for this aim is to identify the recombination rates as well as the recombination hotspots that are present in Bonsmara and Nguni cattle breeds. Now, coming to the materials and methods of the study, we collected data, hair samples from the tail in both Bonsmara and Nguni from a total of 196 Bonsmara cattle breeds and a total of 119 Nguni cattle breeds. DNA extraction, quantification, as well as genotyping was performed at the Agricultural Research Council Biotechnology platform in South Africa. It is very important to note that we used a microarray instead of sequence data. We then went on to perform data quality control using the Plink version 1.9 software rather. Coming to the identification of recombination rates as well as hotspots, in these two breeds specifically, we first did haplotype phrasing using ShapeIt version 2.2. After that, we went on to measure recombination rates in every 0.5 MB windows using the Duo HMM package that is in ShapeIt version 2.2. After that step, we went on to explore the recombination hotspots, and we did that similar to a study that was done by Ma et al. 2015 by measuring the recombination events that were above 2.5 standard deviation in each and every chromosome. Then the identification of genes that are present in all the hotspots regions that we identified was carried out using the NCBI QTL database, bovine QTL database. Tabulated results here show that recombination hotspots in different chromosomes, both in Bonsmara and Nguni, are not necessarily dependent on the high crossover events that are occurring in each chromosome. As we can observe that both in chromosome 1 and chromosome 9, we had a variety in the crossover events, chromosome 9 having the least amount of crossover events happening, and chromosome 1 having the more frequent crossover events occurring in it. However, it can be noted that the total number of recombination hotspots in these two chromosomes was similar, regardless of the total number of crossover events that was occurring in that chromosome. Interestingly enough, it can be noted that chromosome 5, although it had the second least crossover events occurring in it, it tends to have the highest total number of recombination hotspots harboring it. The same can be said in the Nguni data, where in chromosome 18 had the least number of crossover events occurring in it, but had the highest total number of recombination hotspots in it. Chromosome 6 had the highest recombination rate, but it had the second least total number of recombination hotspots, showing that recombination hotspots are not necessarily dependent on the number of crossover events occurring, as well as the frequency at which recombination is occurring. Coming to this graph here, we can further emphasize that the recombination hotspots in Nguni were relatively higher in each and every chromosome compared to Bonsmara, with chromosome 18 having the highest number of recombination hotspots in Nguni, and chromosome 5 having the highest total number of recombination hotspots in Bonsmara. Now, when looking at the nature at which these recombination hotspots are distributed across the chromosomes, we can see that starting with Bonsmara, they are mostly concentrated at the start and end regions of each and every chromosome. When we look at chromosome 8, you can tell the start and end regions wherein the chromosomes are mostly clustered at. Same can be said with the other chromosomes. When we go on to the Nguni data, we can also tell that, in contrary to the Bonsmara data, Nguni recombination hotspots are evenly clustered throughout the chromosome and are not necessarily concentrated towards the start and end regions of the chromosomes. We went on to look at the genes that are involved in these recombination hotspots. I want to draw in your attention closely to these two genes, being the PDE1B gene in Bonsmara and the FP gene in Nguni, which are responsible for carcass traits as well as milk fat percentages in these in Nguni. Now, these breeds are very valuable in the breeding industries as well as the beef industry and the milk industry in South Africa, as these are traits of production and are much needed in most production spaces in our local breeding sectors. Now, what is the takeaway message from this study? We can then observe that recombination patterns are not conserved between species, as it has been previously reported in most livestock species. They tend to vary per breed, as we've noted in our data. We can also conclude by saying that recombination may be used to determine genes that may be beneficial in breeding programs, as we have observed the milk fat protein genes as well as the carcass genes. And these genes are very much beneficial in the breeding industries, particularly in the beef industry, that being the carcass traits. Also, by understanding this, this can then lead to increased production in most beef industries, as we may design optimal breeding strategies that will target the genes that are of interest. In our case, it's mostly production genes. Now, what are the future perspectives from this research? Instead of using microarray data, we can use sequence data. We can further involve more animals, mostly bulls, and we can also use more breeds. Instead of just two breeds being the Bonsmara and Nguni, we can include more indigenous breeds. And that brings me to the end of my presentation. But before I close off, I'd like to give a huge thank you to my supervisors, as well as my lab mates and the funding bodies that were involved in this project, being that the National Research Foundation, the Agricultural Research Council, as well as the RMRDSA, and not forgetting the institution I'm registered in, the Tshwane University of Technology. Thank you very much.
Good morning. My name is Shane Murray, and today I'm going to be talking about the 1K essay project. Our byline is Decoding South Africa's Biodiversity. South Africa is one of the most biodiverse countries in the world due to its species diversity, its rate of endemism, and diverse ecosystems, which includes forests, deserts, estuaries, marine systems, and the world-famous fynbos biome. While it occupies only 2% of the world's land surface area, South Africa is home to 10% of all the plant species in the world, 7% of its reptile, bird, and mammal species, and around 15% of the world's marine species. Many institutions in South Africa research and document local biodiversity. However, South African scientists often conduct genetic research overseas in order to take advantage of competitive prices. This contributes to a drain of skills, data, knowledge, and opportunity out of South Africa. To mitigate this and to build capacity for designing and carrying out biodiversity genomics experiments in country, a South African Biodiversity Genomics program called 1KSA was launched last year. It was launched by Diplomics, which is a genomics, proteomics, metabolomics, and bioinformatics research infrastructure program, which was established by the South African Department of Science and Innovation through their South African Research Infrastructure Roadmap, as shown here. One of the mechanisms that Diplomics uses to enable access to world-class omics infrastructure in South Africa is what we call a high visibility, high impact project. 1KSA, which aims to sequence 1,000 biodiversity genomes in the next 3 years, is one of these. And it aims to build a community to develop different methods for that community and to provide training opportunities in order to increase access of students and researchers to this technology. Under 1KSA, we have 6 different partner labs that are spread around South Africa, and these are listed in the slide. So the Diplomics Training Lab based in Cape Town, the Institute for Microbial Biotechnology and Metagenomics at the University of the Western Cape, Sengen, which is a plant genomics lab based in Worcester in the Western Cape, the University of Pretoria's Genomics Lab, the South African Institute for Aquatic Diversity, which is based in the Eastern Cape. Their aquatics genomics platform is part of the 1KSA project. And lastly, the SAMRC's genomics platform is also involved. Central to the 1KSA project is our website, seen here, which is updated on a monthly basis. We define 4 different milestones for the project, which are listed here. So samples received, samples sequenced, assembled genomes, and then species cards, which are our record of the different species that have been sequenced. This website also provides more information about the project, the DNA requirements for sequencing, the terms and conditions, and also provides links to our Zenodo community, where we list our different methods, our data management plan, and we also have a GitHub link to our bioinformatics workflow. South African-based sample contributors can nominate a species for sequencing under 1KSA by filling out a form on the 1KSA website. This nomination is then considered by the 1KSA steering committee. DNA for the species of interest is then submitted to one of the 6 different labs. All records are kept at every step along the way in REDCap. Whole genome sequencing takes place using Oxford Nanopore's PromethION devices, and a draft genome assembly is generated using the 1KSA pipeline on the National Integrated Cyber Infrastructure System, or NICIS, on their Center for High Performance Computing, or CHPC. Data are moved using Globus Connect and are stored on the Data Intensive Research Initiative, or DARISA platform, which is also part of NICIS. Data are made known via the generation of species cards on the website, and access can be requested through the 1KSA Data Access Committee. So a big component of 1KSA is method development. Nanopore sequencing has a number of different unique DNA requirements, such as high molecular weight, and the requirement of a large amount of DNA as well, and high purity ratios. We are using the ligation and the native barcoding kit, and we've optimized a number of the different steps for library prep, including fragmentation in the instances where the DNA is maybe a little bit too long for those kits. We've also optimized the sequencing parameters in order to get as much data as possible out of the flow cells. And we've also, and it's an ongoing process, that we're optimizing the genome assembly pipeline. So this pipeline includes at the moment quality control steps, a KEMA analysis step, and a nanofield step. We use FLY for the assembly step, and QUAST and BUSCO for the evaluation. We have 1 round of polishing using Recon, which is also then followed by QUAST and BUSCO. This is a draft genome assembly step, and the purpose is to generate some QC data that can be published on the species card. These are some of our preliminary results so far. So on the left-hand side are the read N50 from the sequencing runs, and on the right-hand side are the contig N50 from the draft genome assemblies. And you'll see that both of them have quite a wide range in terms of their kilobase length. And that is, in the case of the sequencing, that is linked to the input DNA that we use. In the case of the contig N50s, we have quite a number of organisms with relatively low contig N50s, but we also were very pleased to find some of them with contig N50s in the megabase range. BASCO scores, we generally obtain BASCO scores greater than 95%, as illustrated here for the 29 plants and 13 fish. We have seen many plants with a high duplicated score, which is maybe not unsurprising, but which we don't see that in fish. And similarly, we don't see that in the 4 mammals and the 3 insects that we've sequenced to date. A major part of 1KSA is also visibility and awareness, and we do this through the generation of species cards, which are published on our website. These are 3 examples. The Buddha's whale, the only example of an inshore whale in South Africa, the speckworm, which is an iconic succulent plant, and the galjoen, which is our national fish. These species card includes information about the distribution of the species, the importance of the species, some of the QC values from the sequencing run and from the draft genome assembly, and also the name of the sample contributor. We also create visibility and awareness through the use of stickers. These are 3 examples for these 3 species here of the stickers that we use. These are very popular at local conferences, always with students. And we also make videos about the different species. Please have a look at the website under stories for some of these videos. I'm now going to talk a little bit about the outputs from our different labs. So Sengen specializes on plants, where they're focused initially on a number of field food crops. They have generated 12 species cards to date. One of the challenges with plants are the very large genome sizes and compared to humans. The South African Institute for Aquatic Biodiversity, their aquatics genomics research platform, is aiming to sequence the fish that are found in South African waters. We have more than 2,000 fish found in South African waters with 75 endemic fish species. To date, 19 species have been sequenced, 18 fish and 1 invertebrate, with many of these fish being on the IUCN lists. At the University of Pretoria, there is a diverse community of scientists studying different species. And some of the species that they have sequenced to date are the Buddha's whale, an antelope, the Cape grosbeak, the Cape fur seal, 2 fish, and more recently a number of fungi, with a number of lessons learned and a number of highlights. At UWC, the University of the Western Cape, the focus is on sequencing bacterial species that they have in their biobank. Their biobank is affiliated to Biodiversity Biobanks of South Africa. And in the initial phase, they are aiming to sequence 500 bacterial species that have been identified out of marine sponges along the South African coast. So the third major component of 1KSA are training opportunities. And we do this through our training partners, through DNA extraction clinics, sequencing workshops, and bioinformatics workshops. And again, just to give you a taste of some of those opportunities, at the University of Pretoria, these are some of the courses that have been run over the last year, including a recent workshop which was allied to the Africa Biogenome workshop for Southern Africa. Sengen's focus has been on plant DNA extraction, and they also launched the field food project earlier this year, which brought in 12 grade 11 learners from local schools to learn about DNA extraction, genome sequencing, and genome annotation. And this is a first for South Africa. At UWC, the focus is on bacterial genome sequencing, and they've also run a number of different training courses. So to conclude, I would just like to acknowledge and thank all the members of the 1KSA consortium as listed here from the 6 different labs that form part of this program. And finally, if you'd like to find out more about Diplomics, please scan this link tree. It'll take you to our link tree. We are very active on social media. Please follow us on social media. Thanks for your attention.
Hello, everyone. I'm Kouame Emmanuel of Nangua Brogwa student in PhD. I'm working in the biodiversity of marine fish of every coast. But today I'm present the era of DNA barcoding in the identification of fish. But so in the context, you can return that the biodiversity is multidimensional and occurs in the variety environment. Ecology diversity, species diversity, and genetic diversity is 3 dimensions of biodiversity. These 3 dimensions is present in all environment, water, area, and vegetal representation. But in the marine biodiversity, the fish is the big class of all specimens present in marine water. So the fish represent 70% of marine biodiversity. But this biodiversity is in the our years. So in the identification of biodiversity, what is the importance of the genetics? The genetics is the basis of evolution. So for understand the adaptation of environment, we use the genetics for this evolution detected and phylogeny relationship C. We use genetics for the identification. We use the genetics and the identification of the new species. But in the marine area, biodiversity is very important. But the recent Côte DNA improve our technique of identification of species identification. But the DNA code is not so the DNA barcoding is not very performant to identification the all diversity of marine fish. That in my presentation I show you, please, when the barcoding is not performant for detect all biodiversity. The barcoding is not performant because in the 7 era of biodiversity of DNA barcoding, and another author say the incorrectly interpretation of code bar biodiversity of code bar DNA is a problem of identification, because the variation interspecific and interspecific is not detected. So in my paper, I make 400 sequence of analyses on the base of fan and all. If I take the all sequence of my fish, I analyze the sequence of in bold by RSL algorithm to make identification. And I can do a ABGD of genomic analyze for the supplementary analyze for the detected species. And I do a haplotype network for see the difference of species. I show you the 3 case study of my fish sampling. In the genus Selen, we can see 3 species. Here, here we can see a Selen peruviana, here Selen setapinis, and here Selen dorsalis. This is a 3 specimen of genus Selen. But in the bold and RSL analyze and ABGD analyze gemic, say this is a one molecular species is present in one bean. But if I do the network haplotype, we can see that the difference of genetic in the 3 species. The distribution of 3 species is so different. We don't say that is the one species. So here, the barcoding DNA is not performant to detect it that we have 3 species in the one genus. The distant genetics of 3 species is 3, but never analyzed can detect it, the difference genetics to see we have 3 species on one bean. Is not the only case. We have a case of SADA, of genus SADA. We are here SADA, and SADA is a different morphologic specimen, but in the genetics, this is the same, because RSL, PTP, GEMIC, and ABGD say that we have a one species in the one bean to, I can tell AAF1389. And another case is the case of genus Pagellus. We have the same problem. The barcoding is not performant to see the difference of 2 specimens who not present in the same area. We have here a Pagellus beloti in West Africa, in Atlantic, and here we have Pagellus nathaniensis that will not present in Atlantic. This is a different specimen of Pagellus. The morphology and taxonomy say this is a different specimen, but the genetics cannot detect it, the barcode gap in the specimen. So here we can see the different genetics. We have a 4-point difference of 2 species, but is not big for ABGD and GEMIC and RSL or PTP. We can see we have a 2 or 3 specimen. Here we see that we have another, we have only 1 specimen, but the haplotype detect 2 group of for the 2 specimen for the 2 species. This is a 3 case when I want to show our, but in my paper I see the problem, the issue of barcoding DNA on the 7 genius in my study. So the barcoding DNA is not bad for detect specimen. In my study, I see that the barcode of Cytochrome oxidase 1 detected 95% of my biodiversity. Is not big because I have 136 specimen. So the DNA barcoding can detect 9.5 specimen. But the barcoding DNA is not performant to detect all species and in the biodiversity. It is dangerous for evaluation. So we can improve and analyze the taxonomy integrative, or we can analyze the molecular gene for another precision. Thank you for your attention.
Hello everybody, thank you for attending to this presentation. So today we will look at a topic entitled Structural Genomics Analysis of the Endocannabinoid System of Bats, that is presented by me, Omar Mashish, and of course my co-authors, Salman Al-Akshi, Walid Bouzi, and Khalid Tawzouji. So we are from the Laboratory of Physiology and Physiopathology of the Mohammed V University of Rabat. As for our presentation, we will start by an introduction to the topic of the endocannabinoid system, which we will follow by a methodology section, the results and discussion, and of course, we will conclude and look at some perspectives. So first of all, let's take a look at what is the endocannabinoid system. So the name suggests it, of course, it's the first thing you will think about when you hear the endocannabinoid system is the plant cannabis or the drug cannabis. So this system was discovered through the effects of cannabis, and we can say that the endocannabinoid receptors are in fact receptors for THC, CBD, and other cannabinoids found in the plants. However, this system is not just restricted to cannabis, it is also a regulatory system. It is the main road policeman of the nervous system, of the immune system, the metabolism, and so on. So the most famous actors of this system are the CB1 and the CB2 receptors, and of course we have the lipidic mediators, which are the 2-acetylglycerol and the anandamides, and the enzymes that make and degrade the mediators, so the Nidal and Aglagane enzyme for the synthesis of the mediators, and the fatty acid amide hydrolase, which degrade them or metabolize them. So this system acts in the nervous system, acts as with a mechanism, a retrograde mechanism, meaning that it potentiates its effects through a retrograde system, meaning it acts on the presynaptic terminal instead of the postsynaptic terminal. So, but it is not just restricted to the brain, it also have other effects, like the control of appetite or the increase of appetite, the increase of craving for palatable foods. It is also linked to lipogenesis, to obesity, to insulin resistance. It has also roles in the immune system. It controls the release of cytokines and other immune mediators. So we can say that it's a very important system that is mostly non-studied, aside from the control of appetite, obesity, and drug abuse. But it can have very good pharmacological actions, meaning that we can use it as a potential symbol for drugs and so on. But our main question here is not about that, it's about what makes the endocannabinoid system of bats interesting. So first of all, we know that bats are very interesting mammals. They live way more than they should. We know that some bats even reach 40 years, and we know that small mammals technically live short lives. Also, the immune system of bats is very interesting. They can tolerate viruses, they can tolerate cancer, they don't even have cancer. And as we know, they fly using a powered flight system, meaning that they should, in theory, make a lot of reactive oxygen species. And we know that reactive oxygen species like degrade the cells and makes the organism more prone to cancer and so on. But bats doesn't highlight those effects, meaning that they have some kind of control over it. And of course, their metabolism is extremely interesting as well. So there is a possibility that the endocannabinoid system could interfere with those processes and make bats the magnificent creatures they are. So we did a small methodology, a small bioinformatic analysis, because this is mainly a preliminary study. It's not like a fully-fledged study, but what we did is that we took the sequence for the CB1 receptor, we aligned it using Mega. We did a motif analysis using MemSuit, the online platform, and we took the promoters, and of course we did a transcription factor binding site analysis using Promo, and we constructed a phylogenetic tree to take a look at the evolution of the CB1 receptor between bats and humans. So the selected species are the vampire bats, the Egyptian rosette, the Cochle's pipistrelle, the Minopterus natilensis, and the horseshoe bat, as well as the humans. We choose those species because they are very well sequenced by the BAT1K consortium, and the genomes are of high quality. So let's take now a look at the results. So when we did the alignment, the first thing we observed is that we have some conserved sequences between humans and the other bats, and we have extremely variable areas in the genome. So the first question that we had was, what are those conserved sequences, how are they, and so on. So what we did is that we did a motif analysis, and we restricted our search to 5 motifs. So the maximum width allowed for the motif was 15 nucleotides, and we did find some interesting e-values. So those are the motifs in the table that is highlighted in this slide. Also, we tried to graphically represent them, and what we can see here is that we have some kind of pattern. We can see that the first motif is always found near the 5' UTR region of the genes, meaning near the start of the gene, and it varies in terms of what follows this motif, but we can see that in the middle of the gene we don't have a lot of motifs, but at the end we have most of the motifs are found at the end, and sometimes they are even repeated, meaning that the closest we get to the 3' UTR region of the gene, the more motifs we find. So this is a point that which we should take a deeper look at. We also did a transcription factor binding site analysis after we extracted the promoters from the sequences. So we can see here in the pipistrelle that we have some very interesting transcription factors, such as the DSF1 or dissatisfaction transcription factor, which control sexual behavior in drosophiles, but will also be the case for bats. We also have the ELK1 transcription factor, which is involved in drug addiction, in light stimulation, in liver development as well as lung development, and also in the apoptotic processes of hippocampal neurons, and by convention to Alzheimer's disease. And we know that bats, when they hibernate, they are in a Alzheimer-like state, but they regenerate after all. Also, we have the homebox A3 transcription factor, which is involved in embryonic development and morphogenesis. So we also took a look at the transcription factor binding site analysis of the Minopterus natilensis bats. We can see that we have some interesting transcription factors as well. such as the state 5-1 transcription factor, which is involved in the response to cytokines, to growth factor, and it is also a mediator of the response of the interleukin-2 and 3. We also have the MYB2 transcription factor, which is involved in cell development and cell cycle progression. Also, the very famous P300 transcription factor, which is a transcriptional co-activator of the histone acetyltransferase family. So it acetylates the histones in the genome for epigenetic purposes. The same could be said about the Egyptian rosette, in which we found the CDX1 or the caudal type home box that activates enterocyte differentiation in the intestines. The BRCZ2 transcription factor, or the broad complex isoform 2, that is a development that is activated during the development and that change under the variation of hormones. Also, when we take a look at humans, one thing that is, we can say, interesting is the vitamin D receptor. So we know that vitamin D is activated under sunlight, and bats don't go in the sunlight. Maybe this is a point that should be further developed. So we did a phylogenetic analysis, and we can see that we have two monophylogenetic groups. The first being the group between the vampire bat and the Egyptian rosette, and the Miniopterus and the Pipistrelle. So this is extremely interesting. Of course, as we know, the vampire bat and the Egyptian rosette, so the vampire bat is a blood drinking bat, and the Egyptian rosette is a fruit bat. So that's a very interesting point. But again, we are taking a look at the evolution of the CB1 receptor, to be specific. So as for perspectives, it will be great to take a look at the biochemical processes that are involved in the endocannabinoid system of bats, and of course to maybe accentuate this bioinformatic analysis. So we can use chromatography, PCR, Western blot, and immunochemistry to take a look at the expression at the level of mediators and so on. And that will give us a bigger picture of the endocannabinoid system of bats. And thank you for being present during this presentation. If you have any questions, please let me know.
Thank you everyone for hanging on there on the biodiversity genomics in Africa. Please, we apologize for the earlier mix-up and the technical glitches we had. And I hope you followed the talk presented very well. Please, if you have questions, there is a Slido provided underneath the streaming platform where you can drop off your questions. Now we're going for the question and answer session. And please, may I remind us that immediately after this session, which should end in the next 15 minutes, hang on, there will be a music interlude, and you can also hang on to join other sessions. We have a couple of questions for the presenters, for the speakers here. The first question goes to Nazifo. The question is, what is the relationship between recombination hotspots and resilience or susceptibility to extinction?
Oh, okay. So there is a somewhat relationship when we take into account the fact that recombination is one of the driving forces of evolution, driving 5 forces of evolution. So we can get ahead with solidifying biodiversity by using these recombination hotspots into targeted selection of genes of adaptability. So we ensure that our species are able to thrive in a given environment by using recombination hotspots to target genes of adaptability to that. So there's pretty a good relationship with resilience to biodiversity of species, I could say.
Okay, thank you, Nazif. You still have another question.
Okay.
How challenging you think it is going to be to aid breeding in cattle largely maintained by the community? Will
Oh, it's not.
rural people join the breeding program? Do you think the rural people would join the breeding program?
Of course, absolutely. They would join the breeding program because in previous years, I remember we had various projects together with this recombinant DNA project where we visited different provinces in South Africa where cattle farmers of rural origins are largely placed. And they were very keen in getting into the program and wanting to advance their cattle in terms of production, in terms of adaptability as well. However, the main key is to make sure that you build trust with rural farmers because what we've noticed mostly with cattle farming is that it's usually the older generation that is into farming. So what helps is having a person who speaks the native language and also explaining to them thoroughly the benefits that the project is going to bring in terms of their livestock, be it in business-wise, commercial-wise, or personally so. So it's not so difficult to get rural farmers involved in such programs.
Okay, thank you for your responses, Nazifo. The next question goes to Sean Murray. Yeah. What criteria do you use to select the species for sequencing?
Sure. So we have a few sort of high-level criteria that are evaluated each time they're nominated. The species needs to be indigenous to South Africa. That's our first criteria. The second criteria is that all the permits for collecting that particular species, for transporting it, and if available, benefits and access sharing as well should be in place as well before we select that species for sequencing. I see in the chat that there's a question about will they be considered if they have potential for breeding? And yes, if they meet those criteria, then yes, we would definitely consider them as well under 1KSA.
Okay, so another question, the second one, the assemblies are always done de novo, or in case you have close related species genomes. Do you use those as reference for the assembly?
Yes, so that's a great question. So the way we've set up 1KSA is that we do a first pass draft genome assembly, which is done de novo. And that is partially to create some QC metrics that we can report on. But we do, as part of the terms and conditions of the program, is that we do give the data back to the sample contributor so that they are then able to run genome assemblies themselves using different pipelines. And so that could be one thing that they might elect to do then, is to use a closely related species in that assembly.
Okay, thank you, Shane. Will I say, fortunately, we do not have questions for Mashish Omar and Kouame Emmanuel. Thank you, every one of you, for your nice presentations. And thank you, the audience, for joining us on this Biodiversity Genomics in Africa. We hope to have a more African country represented in the next conference in 2025. I see that we have more of South Africa and Morocco. Thank you for joining, and I think we are drawing the curtain on this session here, and stay back for the music interlude.